Our next guest speaker is Dr. Jyothi Prabhu. She is the head of the Department of Molecular Medicine at St. John's Research Institute, St. John's National Academy of Health Sciences, recipient of Intermediate Career Fellowship Grant from India Alliance, which is a partnership between DBT and Wellcome Trust in clinical and public health category. She has experience of more than 15 years in oncology-based clinical and public health category. Her current focus is on premenopausal breast cancer, specifically to decipher the interaction between multiple nuclear receptors and their circulating hormones. She is a trained pathologist and later she pursued research as career to gain expertise in molecular pathology and completed PhD in molecular medicine from Rajiv Gandhi University and Health Sciences, Bangalore. Today, her topic is exploring the tumor microenvironment in triple-negative breast cancer, insights for spatial profiling. Welcome, Dr. Jyothi.
Thank you, Swastika, for that excellent introduction. Am I audible?
Yes, you are pretty much audible.
Yeah. I will go ahead to share my screen. Is my screen visible?
Yes. Yeah.
Yeah. Okay. At the outset, I would like to thank Dr. Gopi and the opportunity given by TheraCUES to present this. After an excellent talk by Dr. Kevin, which was so descriptive and a broad overview of all the things that they have been doing and the application of spatial technologies to multiple array of diseases and how the use of AI and ML has been actually brought in to be useful to the patients finally as a translational medicine to bring in some care towards from bench to bedside. I am now going to speak on an important topic, which is very focused work compared to the previous work, which is exploring the tumor microenvironment in triple-negative breast cancer and what we have done using spatial profiling. And in this talk, I would like to talk a little bit about the aggressive nature of triple-negative breast cancer, the heterogeneity of this disease, as well as gains that have been learned using spatial profiling, specifically to understand the tumor microenvironment and how that is useful in strategizing for therapeutic decisions. So to introduce triple-negative breast cancer is a diagnosis of exclusion, and breast cancers which do not have estrogen receptor and progesterone receptor lack the amplification of the HER2/neu are often classified as triple negative. These diseases are very aggressive, they are heterogeneous, and they have a very poor prognosis. Now, as you can see from this picture itself, based on multiple classification system, triple-negative breast cancer is not considered a single disease. It is a very heterogeneous one comprising of multiple subtypes, which have been tried by using multiple systems. These are called molecular classes. And based on the classification system which you would like to follow, there may be 3 to 5 different types, which are often called as basal-like 1, basal-like subtype 2, mesenchymal, and luminal androgen receptor subtype. If you look at the histological subtypes which fall into these classes, these are typically the medullary carcinomas, the metaplastic carcinoma, malignant phylloids, and the ERPR negative invasive lobular carcinomas. One thing that strikes us when we look at triple-negative breast cancer and the Kaplan-Meier curves which appear on your screen at the bottom will clearly show you the aggressive nature of this disease, which is irrespective of the stage at diagnosis. All the triple-negative breast cancers tend to have a poor outcome when compared to the non-triple-negative diseases. The current treatment strategies for this disease, which lacks any kind of target, is using only conventional strategies like surgery and chemotherapy, using both adjuvant and neoadjuvant settings. In the recent past, there has been some interest after the invention of immune checkpoint inhibitors like PD-1, PD-L1 inhibitors. Based on the presence of PD-1, PD-L1, these tumors have also been approved for, at least in the metastatic setting, to be treated by immune checkpoint inhibitors. And also more recently, the more targeted therapy in the form of PARP inhibitors also has been tried for this particular disease. When we come to understanding the tumor microenvironment in triple-negative breast cancer, one thing that becomes very evident when we look at the histological images of TNBC, we understand that there is higher proportion of tumor-infiltrating lymphocytes in these tumors when compared to other subtypes of breast cancer, such as the hormone receptor positive. And the International Working Group on the tumor-infiltrating lymphocytes have done systematic studies looking at the TIL densities. And as you can see in these Kaplan-Meier curves, different amounts of cutoffs have been used by counting these TILs in the stromal area of the triple-negative breast cancers. And they show that tumors which have at least 30% and more of TILs in the stromal area tend to do well, which indicates TILs by themselves are a good prognostic indicator irrespective of the subtype within triple-negative disease. Now that brings us to understand, that actually shows us the importance of understanding the tumor microenvironment, which is a complex and a dynamic thing within any tissue, and especially within a cancerous tissue like a breast tumor. And the tumor microenvironment goes on changing from tumor initiation to tumor progression, and definitely is different at metastatic sites. Initially, when the tumor starts, different conditions which exist within the tumor, such as hypoxia, inflammation, angiogenesis, bring in different kinds of tumor cells as well as the other niche cells into the environment. And there has been, when the tumor progresses, there will be a different interaction between tumor epithelial cells with stromal components and the immune cells, which all contribute to the tumor microenvironment. And thus it becomes very important to understand whether the microenvironment is conducive to tumor growth or is it inhibiting the tumor growth. Of course, in case of triple-negative breast cancer, typically what we know is that it spreads to the critical organs like lung, brain, and liver, which is predominantly responsible for the poor outcome which is associated with this disease. And understanding the tumor microenvironment in that context becomes even more important to identify targets and to improve the prognosis within this disease. Based on at what stage we detect the disease, there can be anywhere from 14 to multiple tens of subtypes of cells within the tumor microenvironment. And that's how it shows how complex it is to understand this particular one. TNBCs are not just heterogeneous in tumor microenvironment or in the molecular subclasses. Even when we look at the genomic architecture, this is a classical study of using more than 160 triple-negative breast cancers from the Cancer Genome Atlas, where they mapped the most common mutations that were observed within this. And as the picture itself tells you, though the TP53 was the most common mutation which is seen in more than half of these cases, there is no single or not even a set of mutations which are common to a set of tumors. And there are multiple kind of mutations, including missense, truncating, indel, splice variants, etc., mostly involving the growth factors as well as multiple proteins involved in the extracellular matrix, such as the laminins, the myosins, the danins, etc. And to add to this, further, there is also a heterogeneity at the immune architecture. Now, typically, based on how the tumor is infiltrated by tumor infiltrating leukocytes, at the histological level, TNBCs within their stroma are classified into three different subtypes. Those which are called as inflamed, which show extremely higher amount of tumor infiltrating lymphocytes within the stroma. Those which are called excluded, which typically show TILs only at the edge of the tumor, and a third category which shows a very poor infiltration with the tumor infiltrating lymphocyte and therefore called as deserted. And within that also, you can see that there is going to be quite a bit of heterogeneity when the stroma itself has a higher amount of TIL versus when the intratumoral component has more amount of TILs. Needless to say that the poorly infiltrated or the desert type of TIL infiltration has a very different subtype of infiltration, mostly consisting of the T regulatory cells as well as the tumor-associated macrophages, which mostly promote the tumor than inhibiting the tumor growth. Now, coming to spatial profiling technologies, spatial profiling is actually an advanced technique which helps in understanding the network between various cells as well as the signaling molecules and the stroma within a disease. We have seen that current biomarkers have limited predictive value to immune checkpoint responses, particularly within TNBC. And spatial transcriptomics as well as proteomics have of late provided new insights into understanding the tumor microenvironment and also in looking at how do we call the responders away from the non-responders to immune checkpoint. And therefore, better understanding of the tumor microenvironment becomes important for personalizing the therapy and also to improving patient outcomes. Now, when we look at the digital spatial profiling technologies, both for transcriptomics as well as for proteomic analysis, we have broadly two kinds of techniques. One which is predominantly image-based compared to another which is more sequencing-based. Now, at the level of detection, the sequencing-based ones often look at the mRNA, which is captured by using barcode beads, whereas the image-based used in situ hybridization as their technology to look at profiling of these tumors. Therefore, the resolution of the sequencing-based is not very great at a single cell level, which is an advantage with the image-based ones. Sequencing-based ones use often array-based capture of the mRNA, whereas fluorescent-based in situ hybridization is the technology behind the image-based ones. The biggest advantage of sequence-based technologies is that it can do the whole transcriptomic analysis in one go, whereas in image-based techniques, the number of measures are limited to a few thousands alone. And when you look at commercial platforms, you have something like Xenium, CosMX in image-based, whereas the Visium and the GeoMX are largely using sequencing-based technologies. Let's look at what is the workflow of digital spatial profiling. As we already have understood, this is spatial techniques are advanced techniques to understand the spatial contexts of cells and how they actually interact within each other. And so necessarily the work begins by looking at the conventional H&E images. And in those areas, you mark what you think is your region of interest or ROIs. These are further enhanced by using morphological markers which differentiate the tumor from the non-tumor or the immune from the non-immune and thereon. And subsequently, the slide is flooded with fluorescently tagged probes to which oligonucleotides are also tagged as a barcode. And then either UV light or laser is shone on these slides to break the bonds of the oligonucleotides in the regions of interest, which are all captured. And the counting of these is done, be it proteomics or transcriptomics, by using either an encounter or a next-gen sequencing technology. The data which is derived from all of this is actually brought back and mapped back to the histological sections from which it was derived. And the data is then compared between different regions of the ROI or different ROIs or a different set of subjects or samples altogether to understand the differences between region to region within a histological section. That's the basis that we are looking at in digital spatial profiling. Now, coming to triple negative breast cancer and various studies which have already shown by doing protein profiling and transcriptomic profiling. I just went through the literature and took out some key studies which have explained how understanding the microenvironment can make a difference to either prognosticate or predict or understand the biology better. One of such study is the one from the Carter et al. Group published in 2021, which looked at spatial immunobiomarkers to predict outcomes and guide treatment in TNBC using a high-plex digital spatial profiling. This showed the importance of spatial microenvironments for identifying prognostic and predictive immunobiomarkers and informed treatment decisions. So it actually led to many such ones, some of which follow here. Another one by Kulasinghe et al. Looked at a small cohort of triple negative breast cancers which differed in their response to treatment, and so they were called as no relapse and relapse groups. And when protein profiling was done between these two groups and differentially expressed proteins separately in the stromal component and in the tumor component were examined, and a bunch of proteins were seen to be differentially regulated in the stroma in the response when compared to the non-respond. So it shows an apt example of how understanding the microenvironment is necessary to even know the response to specific therapy. More examples. This is another example where morphological markers have been very beautifully used to identify the tumor-only component and the stromal component, and the differentially expressed protein composition between the two groups have been mapped, and you can see that there are a set of proteins which are used, and these immune-related proteins within the TME of TNBC were associated with prognosis. More to follow by the Carter group. Recent study by them mapped the intraepithelial and the stromal segments differently in a set of TNBCs which were PD-L1 positive versus the negative, and they showed or identified immune microlandscapes based on PD-L1 expression, and they say that this is how developing immunotherapies and response to therapy is going to move forward in the coming years. Moving on to spatial transcriptomic profiling, some more examples. Transcriptomic profiling has been essentially used to understand tumor biology. One such example is that looking at transcriptomic landscape of breast tumor associated and normal adjacent endothelial cells. This showed that the tumor endothelial cells in the normal and the adjacent endothelial cells have a different set of gene expression within TNBC. And even that shows how important it is to understand the microenvironment beyond just knowing the tumor itself. Also, some more studies which differentiated triple negative based on the races and tried to understand why there have been disparities in outcome based on the race within TNBC. This study used spatial transcriptomics to identify distinct transcriptional clusters and show there are different race-based differences within these clusters. And very recently, from our Indian publication, which has used digital spatial profiling in oral squamous cell carcinoma, they have used the niche between tumor and stromal interface to identify key genes and cell types that differentiated which tumors are likely to go for disease progression and also to predict nodal metastasis. And this could serve as a potential molecular target for oral cancer treatment. Some more studies which were done in understanding how spatial technologies can even become predictive. One such studies is actually looking at tumors which have BRCA1 or 2 mutations, and in comparison to those which are wild type, we see that there is a difference in the pre-neoplastic breast microenvironment, and these markers may help for improving breast cancer risk prediction and prevention in the future. Another study which has done work similar to the oral squamous cell carcinoma, where they have looked at invasive niches in breast cancer, where the study was focused at the tumor advancing edges along with the stroma, and looking at a set of proteins which are listed here, which were labeled as hyaluronine mediated motility. And they show that the specific pathways could be targeted to improve patient outcomes if these signatures are highlighted. All this, I'm sure, actually make you believe that spatial technologies not only help us in knowing what is the tumor microenvironment, but it can also be developed to become either prognostic or predictive markers, and they can be converted into translational studies which can reach the bedside as the technology becomes better and better. Now, coming to our own study, which I would like to present a case scenario of what we have done. And this is based on rising breast cancer incidence in Southeast Asia, where we see that the disease affects more younger and premenopausal women and causes a significant health burden. So the initial objective with which we started was to just compare the reproductive and the clinicopathological characteristics and analyze the molecular subtypes and survival outcomes based on the menopausal status, because we wanted to compare the disease in the younger woman when compared to the older woman. And so this was the study. And towards that, what we did was we did a retrospective analysis of 446 breast cancer patients who were part of a longitudinal follow-up and where we knew the long-term outcome of the disease. This cohort consisted almost more than 1/3 of the patients being younger than 50 years, and most of them were premenopausal. We first compared the tumor characteristics between pre and postmenopausal, and as you can see here, the premenopausal woman had a higher burden of the ER-negative disease, and understandably, they also had higher amount of triple negative as well as the HER2 amplified disease. Further, to know if this has any influence on the disease outcome, we took only the premenopausal breast tumors and subtyped them into the commonly known good performing hormone receptor positive, which is marked here by the green line, and the triple negative, which you can see as a blue line. And of course, it had a poor prognosis when compared to the hormone receptor positive. Now, to know whether the premenopausal or the influence of the premenopausal disease within triple negative breast cancer, we took all the triple negative breast cancer in our cohort and divided them into premenopausal and postmenopausal. And to our surprise, we see that the premenopausal disease actually does much better when compared to the postmenopausal. Now, to rule out if this could be just due to the small number of cases that we have in them, we also performed this analysis on external data sets, which comprised of much higher numbers. This data is from the ScanB cohort, and this is from the Metabric cohort, which has higher number of triple negative breast cancers. And we were able to replicate the similar trends that the premenopausal triple negative breast cancers have, for some reason, better outcomes. The next question that came to us was, is there an influence of sex steroid hormones in these tumors, which is the main difference when we compare the tumors by the menopausal status. To understand that, we performed digital spatial profiling using the NanoString GeoMX Cancer Transcriptomic Atlas in a TMA format on a small set of TNBC samples, which were comprising of both pre and postmenopausal. We selected a total 31 regions of interest, out of which we derived 44 areas of illumination, and we found of the cancer transcriptomic atlas, about 1500 plus genes had a expression in our tumors. Now, taking that ahead, we first performed a cluster analysis only to see that the predominant clusters, there were two clusters, amongst which one was a tight cluster, consisted mostly of the postmenopausal breast cancers, and a wider, probably a more heterogeneous cluster consisting mostly of the premenopausal disease. Now, there are very few postmenopausal AOIs here too, but then they resemble more of the premenopausal than the postmenopausal. Next, to understand if this clustering based on the menopausal status is not either due to the bias or an artifact which is induced by differences in the number of either the tumor cells or the immune cells or the stromal component within these tumors, we performed an estimate analysis and we found that there has been no bias and there has been no difference, therefore, between the tumor purity or the immune score or the stromal component between the pre and the postmenopausal disease. Further, we looked up at the differentially expressed genes in the premenopausal breast cancer when compared to the post, separately in the tumor component, which is of course marked by the pan-CK as a morphological marker, and in the pan-CK negative component. And we found that genes involved in T cell activation, cytokine receptor signaling, and few of the immunogenic processes were all upregulated. Further, we then went ahead to look at differentially regulated pathways to which these genes fall into, and we found some unique pathways in the premenopausal tumors which exhibited immune neutrophil degranulation, interferon signaling, and a downregulation of T cell checkpoint pathways. To understand if this would have an influence on the immune cell type, we did some deconvolution methods to look at the immune cell types separately. And what we found was that there is an increased expression of, at least in a subset of these premenopausal tumors, CD8 positive T cells, endothelial cells, and macrophages infiltration in the premenopausal tumors, indicating that these tumors are characterized by distribution of different immune cell types. To know whether this is further true or not, we performed some immunohistochemistry, and the key insights alone is what I would like to share here. The premenopausal tumors had higher infiltration, stromal expression of CD8 positive cells. We also observed higher intratumoral expression of CD8, CD4, as well as CD20 in the premenopausal tumors. And in addition, the tumors which had higher CD20 intratumorally in the TNBCs were associated with better disease-free survival. Similar trends were also observed for CD4 and CD20, though not statistically significant. Now, what are the clinical implications of this? Now, we know that the disease is not treated differently based on the age or by the menopausal status, be it TNBC or any other subtype. Understanding the tumor microenvironment and also knowing that there could be differences due to specific subtypes within TNBC can be important in bringing out different therapeutic strategies. Of course, our research is actually limited to a small number and has significant limitations needs validation of these findings to explore combination therapies as well as to optimize immunotherapy efficacy within TNBC patients. So, to conclude, spatial profiling has shown significant insights into TNBC heterogeneity based on menopausal status in our study. It can be a crucial factor in understanding TNBC immune profiles as well as in survival outcomes. Personalized therapy approaches based on spatial profiling, and in our case at least, including the menopausal status, could improve the therapeutic outcomes within this dismal class of tumors which often come out with poor prognosis. With that, I would like to thank my team, without which all this work, and the clinical collaborators who were so generous to support us with all the tumors, as well as helping us to build longitudinal cohorts with long-term outcomes, couldn't have been possible. I would like to thank the funding agencies and everyone present here, and to Dr. Gopi and the TheraCUES team for giving us this opportunity. And I would like to stop my talk here and take questions both to Dr. Kevin